Hello everyone, I am Chiranjit and today we will discuss about a very important concept of decoding DNA fingerprinting in an interactive manner. So basically, this technique was first discovered by Dr. Alex Jeffrey in September 1984, whereas in India this technique was first discovered by Lalji Singh. That's why he is called father of Indian DNA fingerprinting. All the human beings worldwide have 99.9% genetic similarity among themselves, whereas only 0.1% variation among them. So let's go a little bit deeper inside the concepts. So basically, a homologous pair of chromosome comprises two types of DNA, that is the bulk DNA, which is 99.9% similar among all the human beings, whereas only 0.1% variation is due to the presence of satellite DNA, which are present in the specific regions of chromosome, that are the telomeric regions and the centromeric regions. So when we unwind the chromosome, we will find that there are many repetitive sequences present in the satellite DNA regions. And these repetitive sequences are in form of tandem repeats. So tandem repeats are nothing, they are the short length of repetitive sequences which are repeated multiple times within a gene. So you can see this repetitive sequences are in the form of tandem repeats. They are repeated in a gene multiple times. It is just like our lockdown routine nowadays, that is eat, sleep and repeat. So the satellite DNA which contains the tandem repeats are further classified into mini-satellite and microsatellite. They are different from each other by following points, that are the mini-satellite are rich in cytosine and guanine bases, whereas the microsatellite are rich in adenine and thymine bases. The mini-satellite, the single tandem repeat consists of 10 to 60 base pairs, whereas in case of microsatellite it is just 1 to 9 base pair. Mini-satellite are also known as VNTR, that is variable number tandem repeats, and this VNTR is used in DNA fingerprinting, whereas microsatellite is also known as STR, that is short tandem repeats. So this is a coding regions where you can see the mini satellite, that is a single tandem repeat of a mini satellite, and this is a single tandem repeat of a microsatellite. So now let's understand the procedure of this technique, in which the specific non-coding sequences of satellite DNA are analyzed to differentiate between individual on basis of polymorphism or variation. So if any DNA sample found at the crime scene as evidence, such as blood sample, swab sample or hair sample, first the DNA is extracted from this sample, followed by the digestion or cutting of the DNA at the specific site with the help of the restriction endonuclease, which are also called the molecular scissor. Now this fragmented DNA is centrifuged to separate the satellite bands according to the density, which is further amplified by the PCR, that is polymerase chain reaction, to amplify the PCR product or the DNA fragment. Now this PCR product containing the fragmented DNA are separated according to their molecular sizes by the process of electrophoresis, that is migration of DNA towards the positive electrode when connected to the power source, which is then transferred to the nitrocellulose paper called Southern blotting. So can you see anything on the nitrocellulose paper? Obviously no. Therefore the nitrocellulose paper is incubated with the radio labeled VNTR probes, which are complement to the respective repetitive sequences. Hence they get hybridized, which is further analyzed by using audio radiography on X-ray plate by hybridized radio label probes. These blots in each lane are very specific and just like our fingerprint. So now let's be a detective and let's solve some cases. This is a case of parental dispute for the property, and both of them claim to be his son and fight among themselves. So let's find out the real son among them. So as we know that offspring receives 50% of genetic material from father and other 50% from mother. So if we need to find the real son, we need to match the position of the lanes, and the maximum similarity among the lanes will be the real son. So in this case, who is the real son? So the maximum similarity is between the parent and the H2, that is the sample 2. So the answer is sample 2. Okay, so now let's go for the Next case. Here we got a hair sample from the crime scene. So can you find out the real thief from the suspect? Pause the video and do it on your own. I hope you got the real thief. Yes, it's the suspect 3. As the DNA lanes of the suspect and the crime scene are exactly similar. So let's put the thief behind the bar. So we are done here. Thank you a lot.